Hello, good evening, students. Welcome to Sankalp Learning Classes. I hope my voice is audible. Yes, already we have started the chapter Biotechnology and Processes, right? So, already we have completed some tools and techniques of the biotechnology, especially some tools about some tools we have studied in the last class. Sorry. Yes. Um, the first tools, that is restriction enzyme, right? So what is the function of the restriction enzymes, and what are all the types of the restrictions enzymes? So that all we have studied in the last class. Then the important tool, that is the vector. So already we have studied about the PBR 322, the vector PBR 322. 322 vector, right? So today, how vectors for cloning genes in plants and animals. Okay, so how the vectors are used to cloning the genes in plants and animals. Here, genetic tools of some pathogens is transformed into useful vectors for delivering genes to plants and animals. So, for example, Agrobacterium tumefaciens, so a pathogen of many dicot plant, and the retrovirus. So, these used as a vectors. Okay, so genetic tools of some pathogens is transformed into useful vector for the delivering the genes to the plants and the animals. So, illy Agrobacterium tumefaciens can deliver a piece of DNA to transform normal plant cells into a tumor. These tumor cells produces the chemicals required by the pathogen. So see here, so this is the Agrobacterium, so tumefaciens bacteria, which has the plasmid DNA, that is the circular DNA, and you can see the foreign gene. Okay, so these tumor cells produce the chemicals required by the pathogen. So when you have introduced this one into the plant cell, so you can see the transformation in the plants. So modified into cloning vector, which is not pathogenic but can use the mechanism to deliver the genes of interest into the plant. So ಕಾಮನ್ ಆಗಿ ಇಲ್ಲಿ ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನಿದೆ that causes the uh, disease, right? So that produces the chemicals required by the pathogen, and which produces the some diseases in plants. So if you remove the piece of DNA, and when you introduce the desired gene tDNA into this, so the plant that can transform, and so here you can see the crown gall disease. Okay, retrovirus. So it is also used in biotechnology. In animals, can transform the normal cells into cancerous cells. So this retrovirus that can transfer the normal cells into cancerous cells, so they are used to deliver the desirable genes into animal cells. Okay, so like this, these both are using in the, so here, which transfer the normal plant into tumors. Okay, that is the TI plasmid. Here, competent host means how to transfer the recombinant DNA into the host cell. Okay, so there are different methods are developed to introduce the, there are different methods which are developed to introduce the transformed cell into the host. Okay, so since DNA is hydrophilic molecule, it cannot pass through the cell membrane. Okay, so bacterial cells must be made competent to take up the alien DNA or the plasmid. So how this bacterial cells they are able to carry this. Okay, for this bacterial cells are treated with specific concentration of divalent cation. So the DNA enters the bacterium through the pores in the cell wall. So here the DNA is hydrophilic. Okay, so because of this, so it is treated with the divalent cation, that is the for example calcium. Okay, calcium has Ca2 plus, so that the DNA enters the bacterium through the pores in the wall. Okay, so that is the thing. So here you can see the transformed bacteria. Such cells are incubated with the recombinant DNA on ice. So these type of these cells, so which should get the some plasmid DNA inside it. So such cells are incubated with the recombinant DNA on ice. So then they are placed briefly at 42 degrees Celsius, that is the heat shock, and put them back on the ice. This enables the bacteria to take up the recombinant DNA. So how this bacteria is able to take up the recombinant DNA? So by this process, that is the heat shock process. So once the incubated, once the cells are incubated with the ice, and once it should treated with the 42 degrees Celsius, so then the bacteria take up the recombinant DNA. Okay, so here also you can see the transformed bacteria. Then some other methods also developed to introduce the recombinant DNA into the cell. The second one is the micro injection. Okay, so here you can see the micro injection. Recombinant DNA is directly injected to the nucleus of an animal cell. So here you can see the how the recombinant DNA is introduced into the nucleus of the cell, that is called as micro injection. And biolistics, it is also called as gene gun. Cells are bombarded with the high velocity microparticles of gold or tungsten coated with DNA. This method is suitable for the plants. Okay, so ಇಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ ಹೈ ವೆಲಾಸಿಟಿ ಪಾರ್ಟಿಕಲ್ಸ್, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಗೋಲ್ಡ್ ಮತ್ತೆ ಟಂಗಸ್ಟನ್ ಜೊತೆ ಈ ಒಂದು DNA ನ ಕೋಟ್ ಮಾಡಿರ್ತಾರೆ. ಸೊ ದ್ಯಾಟ್ this method is suitable for the plants. So this is the gene gun method, where the cells are coated with the tungsten or gold. Then disarmed pathogen vectors. So when it infects the cell, transfer recombinant DNA into the host. So this is the disarm pathogen method. Okay, so these are the other methods to introduce the alien DNA into the host cell. Okay, so these are the some methods. Then processes of recombinant DNA technology. What are the steps involved in the recombinant DNA technology? Let us start this topic. The first step is isolation of the genetic material. Genetic material anna isolate ಮಾಡಬೇಕು from the cells. Okay. Then cutting off DNA at the specific location. So we need particular site of the DNA, particular portion of the DNA, that is called as cutting of the DNA at the specific location. Then amplification of gene of interest using the PCR. So we have only one piece of the desired gene. So amplification means increase the number of the desired gene by using the PCR technique. Then insertion of recombinant DNA into the host cell. Then the recombinant DNA that should be transferred into the host cell. then obtaining the foreign gene product and downstream processing. So we are going to study these all steps in detail. Okay, these 6 steps in detail, that is the process of recombinant DNA technology. Okay, so let us start with the first step, isolation of the genetic material. So treat the bacterial cells or the plant or animal tissue with enzymes like lysozyme, so that is in case of the bacteria, and cellulase, that is in case of the plants, and chitinase, that is in case of the fungus. So treat the bacterial cells or plant animal tissues with these enzymes. The cell is broken, releasing the DNA and other macromolecules like RNA, proteins, polysaccharides and lipids. Okay, see here, you have to treat the bacterial cell. If you are taking the bacterial cell, you have to treat it with lysozyme enzyme. If you are using the plant, cellulase. If you are using the plant tissue, that is chitinase enzyme you should use. Okay, so these will, they broke the cells and release the genetic material along with some macromolecules. Okay, then the second step is isolation of the genetic material. So this is the first step where the isolation of the genetic material takes place. So RNA is, so we have the macromolecules now, right? So we have the macromolecules like RNA, polysaccharides, everything we have. So what we extracted from the plant, animal or bacteria, we have all these materials. RNA is removed by treating the ribonuclease enzyme, and proteins are removed by treating the protease enzyme, and other molecules are removed by appropriate treatments. So we have the samples. So in that sample, all the macromolecules are there, DNA, RNA and protein, and so other molecules also there. When chilled ethanol is added, purified DNA precipitate out as a fine threads as the suspension. So here you can see alcohol onto the sides of two distinct layer of the layers are separated, and spooling of genetic material is observed. So you can see separated DNA can be removed by spooling. Okay, so illy when chilled ethanol is added to this, so you can see the, you can extract the DNA. Okay, then so purified, now DNA is obtained. Okay, already we have the DNA molecule, ನಮ್ಮ ಹತ್ರ ಏನಿದೆ DNA molecule ಇದೆ by this process. Okay, so cells ಅನ್ನ enzyme ಜೊತೆ ಟ್ರೀಟ್ ಮಾಡಿದ್ದೇವೆ. enzyme ಜೊತೆ ಟ್ರೀಟ್ ಮಾಡಿದಾಗ ಏನಾಯ್ತು, we got some macromolecules. To remove all the macromolecules, we use particular enzymes, and only we get the DNA when chilled ethanol is added. So already we have the DNA molecule. Okay, so purified DNA incubated with the restriction enzymes, as a result DNA digest. You know that restriction enzymes, so restriction enzymes function ಏನು, restriction enzymes, so they cut the DNA at the, they make the pieces of the DNA. So they make the fragments of the DNA. Alva, so you know that DNA, length of the DNA, that is 2.2 meter. But the restriction enzymes, they make the fragments of the DNA like this. Okay. Then DNA fragments are separated by a technique called gel electrophoresis. Already we have studied this gel electrophoresis. Here DNA sample we have, so that is treated with the restriction enzymes. So ಇಲ್ಲಿ restriction enzymes ಏನು ಮಾಡಿದೆ ನೋಡಿ, DNA fragments ಮಾಡಿದೆ, and restriction enzymes cleave DNA into smaller segments into various sizes. So ಇಲ್ಲಿ ನೋಡಿ various size ಆಗಿ ಈ ರೀತಿಯಾಗಿ formation ಮಾಡಿದೆ ಅಲ್ವಾ. Then so the gel electrophoresis process, that is used to make the ಸೆಪರೇಷನ್ ಆಫ್ ದಿ ಡಿ ಎನ್ ಎ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್. ಈ ಜೆಲ್ ಎಲೆಕ್ಟ್ರೋಪೊರೆಸಿಸ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ದ ಸ್ಮಾಲರ್ ಫ್ರಾಗ್ಮೆಂಟ್, ಸೊ ಹಿಯರ್ these are the wells where we keep the DNA samples. Okay, so when we apply the electric field, so the smallest DNA molecules, they move towards the anode very fastly, and the larger one, they move very slowly towards the positive side. Okay, so here you will get the separation of the DNA molecules. Okay, so this is actual a gel electrophoresis technique. Okay, so this is the agarose gel used, and you can see the DNA fragments, so when they are observed under the UV radiation. Okay, then the DNA is negatively charged, so it moves towards the anode. So here you can see how the DNA molecules, they move towards the anode. DNA fragments are separated according to their size. Sieving effect of agarose gel. Okay, so sieving effect of the agarose gel. So smaller fragment move faster, and the larger one moves slower. This the process repeated with the vector DNA also. Okay, then can see DNA fragments can be seen as a bright orange color. So these all are DNA fragments, so under the UV radiation, because that is stained with the ethidium bromide. Okay, so like this the DNA fragments are separated. Then second step, so this is the first step, that is the isolation of the DNA, and cutting, second step also completed, cutting off the DNA at the specific locations. Okay, then third step, okay, the DNA bands are cut out from the agarose gel. This is called as elution. The cutout gene of interest from the source DNA and cut vector are mixed and ligase is added. It creates the recombinant DNA. So illi, so this is the gene of interest, so that we obtain from the, so this is the gene of interest obtained from the source DNA, and we obtain this by the gel electrophoresis process. Then and vector are mixed, so this is the vector DNA that we obtain from the host, right? So these both are mixed with the ligase enzyme. So this ligase enzyme that forms the recombinant DNA. So now this is called as recombinant DNA molecule. Okay, now we have the recombinant DNA molecule. So now amplification of the gene of interest using the PCR. So now we have only one copy, right? So we need more copies of this DNA. Polymerase chain reaction. So polymerase chain reaction, PCR, is used in the synthesis of multiple copies of gene of interest in vitro using two sets of primers, the enzyme DNA polymerase. So illi ಏನಾಗಿದೆ, we have only a few copies of gene of interest. Okay, so gene of interest ಏನಿದೆ, only few copies we have. So that we should multiply gene of interest ಅನ್ನ ಏನು ಮಾಡಬೇಕು, multiply ಮಾಡಬೇಕು. So that is the amplification process. So this amplification, this PCR, that involves the three steps. So what is this amplification of gene? So that gene of interest, that is only we have few number. Okay, only one we have, so we have to increase the number of that. Okay, so it has this amplification of the gene of interest, so that has three steps. The first step is denaturation. So denaturation, annealing, and the extension. Denaturation andre ಏನು ಹಾಗಾದ್ರೆ? So just imagine this is the region that should be to be amplified. Okay, amplified. So denaturation means that is the separation of the two strands of the DNA by heating. Okay, so that is the first step. So first step, that is the denaturation. It is the heating of target DNA at high temperature, that is the 94 degrees Celsius, to separate the strands. So in the first step, they separate the two strands of DNA by heating. And the second, each strands act as template for DNA synthesis. So each strand act as a template strand to synthesize the new strand. So like a what you say, replication. Okay. Then second step is annealing. So what is this annealing? So it is the joining of two primers. So here you can see the RNA primer that is joined in this, and here also you can see the RNA primer. So at the 3' end of the DNA templates, it is the joining of the two primers. Here you can see one primer that joined the 5' to 3' template strand, and another primer is also added at the 3' to 5' strand of the RNA. Okay. Then the third step is extension. So here addition of the nucleotides. So the nucleotides that is added by the DNA polymerase called the Taq polymerase. So that is isolated from the thermus aquaticus. Okay. So which add the nucleotides towards the 3' end. Okay, so towards the opposite end, right? Like it synthesized the new strand in the 5' to 3' direction, and here also 5' to 3' direction. Okay, so the amplified fragment can be used to ligate with the vector for the further cloning. So here the amplified, so here we will get more copies of the amplified DNA in this process. So that is the gene of interest. So what we have taken for the experiment, that is the gene of interest that can be used to ligate with the vector for the further cloning. Okay, so this is the third step. Then so PCR at glance. So again you can see denaturation, separation of the DNA strands, and annealing is attachment of the primers at the both the ends of these strands. Both are acting as a template strand. Extension means the DNA polymerase that adds the nucleotides that synthesize the new strand. So here from one gene of interest, that is going to form the two copies of the new copies, two new copies. So here, so in the PCR, you will get the billions of copies of the DNA from the single gene of interest. Okay. Then the fourth step is insertion of recombinant DNA into the host cell or the organism. So now we have the recombinant DNA. So recombinant DNA andre, so this is the which is having the gene of interest. Okay, so this is the plasmid which is having the gene of interest. Using any methods, the ligated DNA is introduced into the recipient or the host cell. So here E. coli is the host. Okay, so this recombinant DNA that is having the gene of interest, recombinant plasmid which is having the gene of interest, that is transferred to the E. coli. Just imagine that is the host. If recombinant DNA bearing ampicillin resistant gene is transferred into the E. coli cells, the host cells become ampicillin resistant cell. So ನೋಡಿ ಇಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ ಅಂದ್ರೆ recombinant DNA. So if this recombinant DNA that is having the ampicillin resistance gene, so ಇಲ್ಲಿ just imagine ಮಾಡಿ ampicillin resistance ಮಾಡುವಂತ gene ಇದೆ ಈ ಒಂದು segment ಅಲ್ಲಿ. So the host cells become ampicillin resistant. So this bacteria now is able to resist the ampicillin because of the presence of this ampicillin resistance gene. Okay, so if the transformed cells are spread on agar plates containing ampicillin, only transformants will grow, untransformed recipient cells will die. So illi, so if you grow this bacteria in a ampicillin containing nutrient media, the transformed cell will grow, the non transformed cell they will not grow. Okay, because of the ampicillin resistance, ampicillin antibiotic. So even the transformed bacteria ಏನಿದೆ ampicillin resistance ಇದೆ, so it will grow. So this non-transformed cell that will not grow. Okay, so that is the insertion of the DNA into the host or the host cell or the organism. Then the fifth step is obtaining the foreign gene product. The ultimate aim of recombinant DNA technology is to produce a desirable protein. So now the recombinant DNA that is present in the host cell, right? Here, if a protein encoded foreign gene is expressed in a heterogeneous host, it is called as recombinant protein. If the protein is able to produce in this process, so that is called as the desirable protein. So we are getting the desirable protein in this process. Cells with the foreign gene can be grown in a laboratory. The cultures are used to extract the desired protein and purify it with using the separation techniques. So already we have introduced the recombinant DNA into the host cell. Now it is able to produce the desired proteins. Okay. Now the host is able to produce the desired protein. A protein production ಆಗ್ತಾ ಇದೆ. The cells can also multiply in a continuous culture system. Here, the used medium is drained out from one side while the fresh medium is added with from the other. It maintains the cells more physiologically active and produces larger biomass. So it yields the more desired protein. So now we need the desired protein that is obtained from the host cell. Okay. So these are called as the bioreactors. So these are the vessels in which raw materials are added, biologically converted into specific products, enzymes, etc. using microbial, plant, animal or the human cells. So if you put the all raw materials in this, so the enzymes products, so using microbial or plant or animal human cells, bioreactors are used to produce the large quantities of products. They can process 100 to 1000 liters of culture. So illenagide andre, so in a lab, so to get the desired gene product only, that is limited, it is not possible to get in a large quantity. So large quantity ಅಲ್ಲಿ ನಮಗೆ desired gene product ಬೇಕು ಅಂದ್ರೆ, so the bioreactors are used. So they can process 100 to 1000 liters of culture. Bioreactors provides optimal growth conditions, temperature, pH, substrate, salt, vitamin, oxygen to get the desired product. It is providing the optimal conditions to get the desired products. Okay, so that is also helping to obtain the desired gene product. Here, bioreactors, most commonly used bioreactors are stirring type. So, stirred tank reactor. Here you can see simplified stirred tank bioreactor. So, this is the simplified bioreactor. It is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. So if you put all the essential raw materials inside this, so that helps to get mix this all the constituents raw materials in this. So here you can see this is the simple stir tank bioreactor. The stirrer facilitates even mixing and oxygen availability. Alternatively, air can be bubbled through the reactor. So here you can see the air, so that is coming out, so the bubbles formation, so in the reactor, bioreactor. So this is the diagram of the both how it takes place. Okay, bioreactor, how it works. Then the last step, that is the downstream processing. So what is this downstream processing? It is the series of processes such as separation of purification of the products after the biosynthetic stage. So downstream processing means some series of processes that helps in the purification of the products. Already we obtained the desired genes to purify that product. So this downstream processing is used. Okay, the product is formulated with suitable preservatives, such as formulation undergoes the clinical trials and strict quality control and testing. So ಇಲ್ಲೇನಾಗುತ್ತೆ, ನಾವು ಪ್ರಾಡಕ್ಟ್ ಅನ್ನ ಏನು ಆಬ್ಟೈನ್ ಮಾಡಿದೇವಲ್ವಾ, ಸೊ ದಟ್ ಶುಡ್ ಅಂಡರ್ಗೋ ಸಮ್ ಕ್ಲಿನಿಕಲ್ ಟ್ರಯಲ್ಸ್ and strict quality control and the testing, then only it is allowed to the market. Okay, so that is called as the downstream processing. Okay, so these all are the steps involved in the uh, recombinant DNA technology. Okay, so here you can see the downstream processing. So bioreactors and obtaining the foreign gene material, amplification of the isolation. So these all steps we have completed. The first step is isolation. We have isolated the genetic material. First thing is we treated with these all with the enzymes to get the macromolecules like DNA, RNA, proteins and all. Then we treat with this with the some protease and the ribonuclease to remove the RNA and protein to remove these all materials, and we will get the DNA. So that is the genetic material we will get. Cutting off the DNA at the specific location. So then comes to the restriction enzymes. They cut the genetic material into in the particular site and make the fragments of the DNA. Then, so if we get this desired gene, so just imagine this is the desired gene. So I need more copies of this desired gene. Now I have only one copy. I need more copies of this desired gene means, so we use the technique PCR. So that is the polymerase chain reaction where the three steps are there: denaturation, annealing, and the extension. So we can get millions of copies of the desired gene, and you can insertion of the recombinant DNA into the host cell. So cutting of the DNA and insertion of the desired gene into the plasmid DNA to form the recombinant DNA, and insertion of the recombinant DNA into the host cell. So that recombinant plasmid that is introduced into the host cell. Host cell ಗೆ introduce ಮಾಡಿದ್ವಿ. So that is the E. coli cells. So we can see the transformed cell and non-transformed cell by growing them in the ampicillin media. Okay, so when they grown in the ampicillin media, we can obtain the foreign gene product from that. For example, desired proteins and all we can obtain, and the downstream processing. Okay, so these all are the steps involved in the recombinant DNA technology. Okay, one of the important topic so that we have covered today. Then, so these are the methods how to introduce the recombinant DNA into the host cell. How we can introduce that recombinant DNA? So, microinjection method, biolistic method, and disarm pathogen. So, where the some disarm pathogens are introduced, used to introduce into the host cell. Then, one more important one is, so here to make the to introduce the plasmid into the recombinant gene into the host. So the bacterial cell is treated with the divalent cation. Then it should be treated with the ice and again heat. So then the bacterial cell is able to get this recombinant DNA. So this is called as the transformed bacteria. Okay, so these all are the different methods that is used to introduction of the DNA into the host cell. Then, so here this how the vectors for cloning genes in plants and animals. So this agrobacterium tumefaciens, that is the plasmid is used to deliver the genes inside the plants. So that is called as ti plasmid. The disease causing mm, the gene is removed and the desired gene is introduced, then we can introduce that into the cell. Okay, so that is the method. So these all things that we have studied in today's class. So just recall what we studied in the last class. So that is about the, just I'll show. So this PBR 322. Okay. So features of the very important, I repeat this once again, the features of the cloning vector, right? So which has the different uh, sites, that is the origin of replication, ORI. So that is the where the replication begins. Then marker gene, selectable markers. So selectable markers means, so we can select the recombinant and non-recombinants. So why I'm repeating this topic? Because so we have used the transformed and non-transformed bacteria. We have selected that in the recombinant DNA technology. So that is based on the ampicillin resistance and the tetracycline resistance gene present in the uh, that vector. Okay, so these are called as the selectable markers. Then cloning sites. So in the PBR 322 vector, you can find the different cloning recognition sites like PST1. So the enzymes are able to remove that region and able to attach the desired gene in that region. Okay, so that is called as the cloning sites. So we can obtain the transformed and non-transformed uh, bacteria by using this one clone. Okay, so this is how transformed bacteria and non-transformed bacteria that is recognized in the medium. Okay, so the transformed bacteria means which is have the recombinant DNA that is able to grow. If the ampicillin gene is present in that one, is able to grow in the ampicillin medium. So if it is having the tetracycline resistance gene, means that is able to grow in the tetracycline medium. Okay, so that is the about the vector. Okay, yes, so these all the topics that we have studied in the last class. So I hope this class is very useful for you people. If you have any doubts, dear students, just write in the comment box. Okay, thank you. We'll continue in the next class.